Well, good day, everybody. This is Chris Bear, and I'd like to welcome you back to Biology 225, Section D08, Human Anatomy and Physiology 1. We're a little over a week away from our first class, and I wanted to give you guys a couple of tips, hints, what have you, to get you better prepared for your first class, because your first class is actually going to be lab as opposed to lecture. Again, I outlined this on the first video that we would begin on January 21st, 2016 at 5:30 in the lab. You can find that on the syllabus that has been attached to your homepage on Canvas. And if you're seeing this video, if you're watching this video rather, then you are able to get into Canvas successfully. So the labs, the way all of the labs are set up is there will be a pre-lab or preparatory component to your lab exercise. So for your lab manual, what I've done is I've actually copied the first exercise, and I'll just kind of demonstrate where you find those. It's actually pretty self-evident, but I figured I'd do that anyway on this first lab just to give you a heads up. So what you're going to want to do to be better prepared for lab is you're going to want to make sure that you've done the reading, okay, in your textbook that corresponds with the material that we'll be covering in lab. Again, lab and lecture are not separate entities in this particular course. They're complementary, okay? Lecture complements lab, lab complements lecture, and so there is significant overlap in the material, and the lab experience, again, is designed to complement lecture, lecture is designed to complement lab, and that's kind of how things work in science in general. To really understand something, yes, you can sit in a class and you can go through a lecture, but then a big part of the scientific method involves doing experiments, involves going into the lab, involves applying the information that you learn in lecture, because it's really easy to kind of get into this way of thinking where, oh, I heard this from somebody in a science class, and they're a professor, instructor, what have you, and they said this, a kind of a sage on high type of situation, if you will. Okay, that's all well and good, but how do we know the things that we talk about? Okay, how do we know that they're actually a reflection of the real world? Well, we go into the lab and we go, okay, this is what we talked about. Okay, this is what the book says. This is what a lecture said. Okay, let's see how that pans out, and we do experiments. In the case of anatomy and physiology, we'll do dissections. We will look at cells. We will set up different experiments and look at tissue samples and look at models of the human body and make those distinct connections. A big part of this course, I want to make it less about me standing up there and saying this is the way the world is, and rather what I want to do is be able to provide you a foundation of knowledge, and then we'll go out into the lab and we will actually see how that holds up, and we should be able to bring both the theoretical, if you will, the didactic and the practical part of science, and kind of bring those together. So with that in mind, your first lab is going to be Chapter 1, that's Introduction to Anatomical Terminology, and then Chapter 2, this is in your lab manual. Chapter 1 and Chapter 3, Chapter 1 and Chapter 3 in your lab manual. Chapter 2 is the chemistry. We won't be doing the chemistry, or you won't have a chemistry lab report to do per se, so you won't be required to do that. There will be some chemistry exercises that I'll demonstrate, but just due to time constraints, I'm not going to be able to do very detailed, a very detailed or aggressive chemistry lab with you guys, but one of the requirements for this course is either concomitantly or before you finish the year sequence of anatomy and physiology is that you take either Chemistry 110G or Chemistry 111 with a lab. So if you look at the lab manual, what you'll see is you'll see generally at the very beginning, you'll see something that says pre-lab, there we go, pre-lab exercises. And I've actually gone through all these exercises and found the answers and filled them in. So I actually do a lot of this stuff as well. So what you'll do is you'll want to do the pre-lab exercises, and so you just go in, okay, you find the answers to the questions, all the pre-lab stuff, and then when it says exercises, okay, that's actually stuff that we'll do in lab. We won't necessarily do every single exercise for every single report, and I will let you guys know which parts of the exercises I want you to do and which parts I don't. And generally what I'll do is when we come into lab, I'll spend a little bit of time, if you want to call it lecture, but I'll spend a little bit of time priming you or prepping you for the lab, just doing an overview of the material, and then there may be some demonstrations that I'll do. For example, in this first lab, we will be doing some dissections. We'll be dissecting a fetal pig. And there I will demonstrate the technique, and I'll point out significant anatomical landmarks that will prime you for when your group goes in and you are actually doing the dissection. Then you'll have an idea of how to do it, what structures you're looking for, and so I'll do things like that. But it's real important, if you do those pre-lab exercises, you're going to have them done anyway for the lab report. You might as well do them before the lab. That primes you, that preps you. You're thinking about things already, and that'll make the actual exercise a lot easier. In addition to that, we'll be doing exercise 3. Okay, so 1 and 3 will be in the first lab, and that is introduction to the microscope. And the introduction to the microscope does not have a pre-lab exercise. It just starts on the exercise. So this first lab is going to be fairly straightforward, not particularly robust. And the only pre-lab exercise you're going to have will be in part 1, introduction to anatomical terms and terminology. But if you do those pre-lab exercises, that again, it primes you, preps you, gets you thinking about things, and it should make your overall lab experience go by a lot easier, and you hopefully get a little more out of it. For this first lab, I will spend just a little bit of time going over the syllabus. I will talk about some study tips for you guys as far as reading goes, specifically reading your textbook, some tips and some things that will help you get through the material. I'll take any questions, and then we'll roll right into the lab. I will talk briefly. I'll do an overview, because this will be your first exposure potentially with human anatomy and physiology. I will do a little bit of lecture. I'll get you primed as to what anatomy and physiology is, some of the things that go into it. We'll talk about some of the terminology. I'll point some things out. Okay, and then we will roll right into the first lab. I'll do a demonstration on the fetal pig, and then you guys will quote unquote run amok, if you will, in groups, and I'll be there monitoring your progress, and I'll be available to hopefully facilitate learning, answer any questions you may have, point things out. I may even ask you questions as you work through the lab. And then once we finish with the anatomical portion of the lab, we'll move on to the microscope. This particular lab is going to be a familiarization with the microscope. I will talk just a little bit about the history of microscopy, in general, how it works, the properties of light, and I may even mention some of the other techniques that are utilized, microscopic techniques, most of which we actually won't be using in human anatomy and physiology. Certainly not more advanced, like atomic force microscopy and scanning tunneling, electron microscopy, those particular modalities, fluorescence microscopy as well. But we will do some limited. We have some limited exposure with staining. Those of you guys that are going on into nursing, respiratory therapy, diagnostic sonography, dental hygiene, dental assisting, let's see here, x-ray technology as well, radiographic technology, those specific nursing and allied health disciplines, you will have to take microbiology, and in microbiology you'll actually spend more time staining, using stains, gram stain. You'll talk about acid fast staining and different techniques that we can use more for identifying bacteria, but the same kinds of the same motivation applies in human anatomy and physiology, where we can use different stains to enhance the tissues or the cells that we're looking at. And some of the tissue slides, as we move into cytology, or the study of cells, and as we move into histology, or the study of tissues, many of the slides will already be pre-stained for you guys, so you won't have to worry about the actual staining procedure. But the fact that we have these procedures available is still important. And then we'll move into looking at the microscope, learning how to just set it up. We won't be looking at any specific types of tissue or cells for this first lab. It's just going to be very small. We'll have very small letters that you'll look at. You'll be able to focus in on them, learn how things work, increasing, decreasing light levels. These are what we call compound microscopes. So the islands, the eyepiece has a certain magnification, and then you have different lenses that you will rotate, and we'll talk about those, and they have a different magnification, and the magnifications kind of add together. They compound, if you will, hence the term compound. And we'll talk about how we get total magnification out of the two different lens systems set up. But we'll talk about all that in lab. So that's just an introduction to your first lab. Be actually a really nice way to ease into anatomy and physiology. And the really cool thing about doing lab first is once we go into lecture the week after that, that Tuesday, you will already have a significant portion of Chapter 1. The material that we cover in Chapter 1, you will have already covered in lab. You've already gotten your hands dirty. You've already started to use this terminology. And so Chapter 1 will be relatively easy. We'll be able to blast right on through that, and then we'll start in on chemistry, and we'll see how far we get through the chemistry. It won't be an incredibly robust lecture on chemistry, simply due to the fact that I have so much material to get through. But chemistry really is it's the foundation of biology, and I'll talk a little more about that when we get into lecture, about where chemistry, even physics, really physics. We talk about physics as a scary word, but boy, oh boy. Virtually everything reduces back to physics, and specific applications of physics are where we get different fields. And we'll talk about that in more detail. But really, chemistry is fundamentally applied physics. We apply principles of physics, and that explains chemistry and chemical bonding. And molecular molecules and all the energy and the bonds in that involved in that. It's all due to an application of physics, and then applications of chemistry lead into biochemistry and different forms of chemistry, and eventually lead us to biology, which is where human anatomy and physiology kind of comes out of biology. So if you take it back, it all kind of goes back to physics. So it's important stuff. We'll talk about it in a little more detail when we get there. Alrighty, guys. Hopefully you found this helpful. So yeah, please don't wait to get started on those pre-lab exercises. It just saves you a lot of time. It'll allow you to get out of lab at a reasonable time. It'll allow you to get your report in. Remember, your reports are due at the end of lab. So you got to get those reports in. And even though they're only worth 13.5 points a lab report. Over the whole semester, collectively, your lab reports are about 15% of your grade. So that's not a minuscule part of your grade by any means. That's a difference between an A and a B, a B and a C, C and a D, right? That's 15%. So certainly it's an important part of your grade. So there you go. Hopefully this was helpful, and we'll see you guys on the first lab. Take care.